Hello students, today we are going to start a chapter, microorganism. Now, what are microorganisms? The world is divided into two wings, living beings and non-living beings. Now, living beings are of different types. They are found in air, water, soil, everywhere, okay? Now, they are animals that live in deep in oceans, in streams, in polar regions, even in hot deserts, cold deserts, everywhere. These types of animals are living there. This soil, the many living beings which we cannot see from our eyes, and these beings are called microorganism. The soil which we have, if you gather that soil, a single particle of that soil will be having 50 million types of bacteria in that. What are bacteria? Bacteria is a type of microorganism. So some microorganism like bacteria, protozoans have a hard outer covering called cyst. Okay, now in favorable condition, they emerge out from the cyst and multiply and continue their life cycles. The study of microorganism is called microbiology. That is, micro means small and biology is divided into two words. Bio means life and logos means study. So, the study of small life is called microbiology, okay, or the study of microorganism is called microbiology. Now, we will discuss all about this in our chapter. Now, what we will be studying in this chapter is, we will be discussing about types of microorganisms, that is bacteria, algae, fungi, protozoas and viruses in detail. Then we will be discussing about, the common harmful effects of this microorganisms, and we will be discussing about antibiotics, vaccines related, and how these microorganisms are useful to us. We will discuss all about this in our chapter. Now, let's start with the main heading, study of microorganism. Study of microorganisms. Okay, this microorganism we cannot see from our naked eyes. They are small, they are very extremely small. They are even small as 0.0001 mm. That is, if you have a scale and you have 1 centimeter, try to divide that 1 centimeter into 1,000 parts and try to see that. This small animal, that small organism can be there, and this organism is called microorganism. Long ago in 1677, in which year? In 1677, in June, when Leeuwenhoek, he was a Dutch scientist, he was looking at a drop of pond under a crude self-made microscope. What is microscope? Microscope is an instrument which is used to see objects which cannot be seen by our eyes. So, he had made a crude self-made microscope and he watched a drop of water of pond. And what did he see in that drop of water? But then he sees some tiny living organism there, and these tiny living beings were moving everywhere. They were moving everywhere. And then this tiny organism, this A.V. Leeuwenhoek named this tiny organism as microorganisms or tiny organism. We call it tiny organism or microorganism because micro means small. So that's how we come about to know the existence of microorganism. And after this also, इसके बाद भी बहुत सारी discoveries हुई हैं कि हमें other types के microbes पता लगे हैं. Microbes को फिर हमने classify किया है. फिर इनके इम्पैक्ट्स के बारे में जाना है हमारी ह्यूमन लाइफ पे. सो वी हैव डिस्कस्ड एवरीथिंग अबाउट एंड वी हैव स्टडीड एवरीथिंग अबाउट दिस माइक्रो ऑर्गेनिज्म. इवन नाउ अभी भी हम माइक्रो ऑर्गेनिजम्स के बारे में जान रहे हैं, इनकी स्टडीज़ हो रही हैं, रिसर्चेस चल रहे हैं, जिससे कि हम जानते हैं कि हमारे बॉडी पे या हमारी ह्यूमन लाइफ पे इसका क्या क्या इम्पैक्ट होता है, आइदर यूजफुल और आइदर हार्मफुल. Microorganism can be good also, microorganism can be bad also. So like humans, they are good also, they are bad also. Okay? So now there is an activity given in your book regarding that is how you can see microorganism in pond water. So just simple, collect some water from a pond, take a microscope and try to see that. Take a water from the pond, clean it, that's all the impurity, that's dust particles, all the small sticks, grass should be filtered out. Take a drop from that filtered water, put it on a glass side, see it under microscope, you will be seeing some moving animals under it. So these are microorganisms. Now, microorganisms can be classified into 5 categories. As you can see in the chart given in your books about the classification of microorganism, that is bacteria, algae, fungi, protozoans and viruses. So this bacteria, the examples of these types of microorganism is given, that is in bacteria streptococcus, chlorea and lactobacillus. In algae oscillatoria, volvox, blue-green algae. In fungi, yeast, molds, mushrooms. Yeast, mold, mushrooms are used by you. You know mushrooms as you use mushroom as a food also. It's a fungi. Yeast is, you love to eat breads. Breads are made with the help of yeast. Okay. Protozoans, amoeba, parasemium, plasmodium. And the example of viruses, AIDS, polio, chickenpox. Viruses are the most deadly type of microorganism because mostly viruses are bad. Mostly, okay? Not every organism is bad, but microorganism, the virus category of this microorganism is mostly in a category that they always try to have a harmful effect on us. Now, we have many scientists who have contributed to the field of microorganism. So A.V. Leeuwenhoek first established the existence of bacteria. He was the first person जिसने हमें बताया था कि bacteria exist करते हैं. Okay. Next is Louis Pasteur. He was famous for his discovery of fermentation and pasteurization. You all know about this fermentation, that is wine. How this wine is made? It is made by the process of fermentation. And what is pasteurization? This milk, you have, maybe your family will be bringing a packed milk, that is they will be milking Saras milk or they will be getting a milk of Amul. Please see on that wrapper, on that packet, it will be written as pasteurized milk. What is pasteurized milk? So that it became free from microorganism and do not get spoiled easily. And that's why he was called father of bacteriology. Okay. Now, F.J. Cohen, he studied about bacteria in detail under a compound microscope. There are different types of microscope. There are simple microscope, compound microscope, electronic microscope. So he studied about this bacteria in detail under compound microscope. F.J. Cohen. Now, Dmitry Ivanovsky, okay, he was a Russian scientist and he first discovered viruses in 1892. In which year he discovered viruses? In 1892, he discovered first viruses. Now, viruses have a different type of structure than other microorganisms. Every microorganism is different from each other. Next scientist is W.M. Stanley. Now, he first discovered isolated tobacco mosaic virus in crystalline form. Okay, he discovered a virus called tobacco mosaic virus in crystalline form. Next is Edward Jenner. He played a major role in vaccination. You know, vaccination, you all will be hearing some news about this COVID-19 situation, and you will all be like, everyone is in a hurry to create a vaccine, and this process of vaccination or this vaccination, the importance of vaccination or how this process of vaccination was developed, it was given by this Edward Jenner. So we should be proud of this. All the scientists who have given their contribution to this field. And the next scientist is Alexander Fleming. He discovered penicillin. Penicillin is a type of antibiotics that is developed from microorganisms. Okay. Next is Sir Ronald Ross. He reported that malaria parasite is transmitted through Anopheles mosquitoes. So that was a person who had told us that Anopheles mosquitoes, Anopheles maybe bacha, female Anopheles mosquitoes transmit malaria in our human bodies. So next we will be studying about bacteria. Okay, all the 5 microorganisms we will be discussing each and every one in detail, and the next one that we will be discussing is bacteria. So this was about study of microorganism. हमने हमारे अलग-अलग साइंटिस्ट के बारे में पढ़ा जिन्होंने माइक्रो ऑर्गेनिज्म के बारे में हमारे बताया कि माइक्रो ऑर्गेनिज्म कैसे होते हैं, उनका इम्पैक्ट क्या होता है हमारी लाइफ पे. नाउ द नेक्स्ट क्लासिफिकेशंस, बैक्टीरिया. Bacteria are very different types of bacteria are there. Let me write. Decomposers, aerobic, anaerobic. Okay, now decomposers, these bacteria, they feed on human feces or animal feces, waste material, and eat them as their food. These are called decomposers. Bacteria decomposers. Okay. Aerobic, the bacteria which grow in the presence of oxygens are called aerobic bacteria. Okay. And the bacteria which grows in the absence of oxygen is called anaerobic bacteria. Okay, these are 3 different types of bacteria. Other than this, bacteria can be classified on the basis of their shapes also. So the next thing we will be discussing about bacteria is the shape and structure of bacteria. The first shape and structure of bacteria can be coccus or round shape. First, coccus or round shaped. These bacteria have spherical shape, okay? This is why that is called coccus. Coccus is a singular word. Plural word of coccus is cookie, okay? This is the plural word. And the example of this coccus bacteria or the round shaped bacteria is streptococcus, and this streptococcus, it causes pneumonia, the disease. आपको pneumonia अगर हो रहा है तो आपकी body में कौन सा bacteria होगा? It will be streptococcus. The bacteria streptococcus causes pneumonia. Okay, so this is coccus or round shaped bacteria. The next shape or the next type of bacteria according to shape and structure is bacillus or rod shaped. What is? Bacillus or rod shaped bacteria. bacillus or rod-shaped bacteria have a shape in a form of spiral or a rod. Okay, and this bacillus or rod-shaped bacteria, they have, or they may not have a flagella. What? Flagella. You know what is flagella? Suppose this is a bacteria, and this bacteria, if it has this strip, this is called flagella, and it helps into the movement. So some bacteria may have this flagella and some may not have this flagella. So this is, so bacillus bacteria may have or may not have flagella in them. Okay, and you know this bacillus bacteria, they cause many types of dangerous diseases. You know, tuberculosis, diphtheria, tetanus, leprosy, all these are called by bacillus types of bacteria. Okay, so it is very dangerous type of bacteria to us. Okay, so you know, Bacillus typhosus. What is Bacillus typhosus? Let me write down. Bacillus typhosus, or the next name of Bacillus typhosus is Salmonella enterica. Okay, enterica. This bacteria causes typhoid. So, you know, this bacillus causes tuberculosis, diphtheria, leprosy, tetanus, and also typhoid. The next type of bacteria that is divided on the basis of shape and structure is spirillum or vibro-shaped. Okay, next is spirillum or vibro-shaped bacteria. Now, these bacteria are in a form of spiral, that is, this type of spiral type shape is there of this type of bacteria, or a comma shape. You can say comma shape or corkscrew. No, corkscrew is, this is, you have seen a nail, and on it nail this type of structure is given. So, this is called corkscrew. So, the shape of this bacteria is in corkscrew shape or comma shape. Okay, and also this bacteria may have cilia, one or more cilia or flagella may be there on this spirillum or vibro-shaped bacteria, and the example of this bacteria is Vibrio chlorea, and which causes chlorella disease. You can see in your books the image of all these bacteria under microscope is given in your books. So, please refer to that book images. The next activity is given in your book regarding the effect of temperature on bacterial growth. Now, just take 3 bowls and take milk of 3 types: hot milk, warm milk, and cold milk. Add a little amount of yogurt to each of the bowl and keep it aside. Mix it well and keep it aside for 5 to 7 hours. You will be seeing that in hot milk, no change occurs. In warm milk, you will be seeing that the milk is converted into yogurt or curd. And in the cold milk, you will be seeing that the milk will be converted into curd, but it will be taking a longer period of time. So, you know, the bacterial growth, the proper temperature for the bacterial growth is warm. That is, they do not require very high temperature to grow, or they do not require very low temperature to grow. So, this was the activity given in your book. Now, the next heading that we are going to study about the relation of bacteria, that how these bacteria are useful to us. So that is the heading, uses of bacteria. So the heading is uses of bacteria. Now, bacteria. You know, the first use that is given in your book is nitrogen fixation. What is nitrogen fixation? As I have told you about Rhizobium bacteria, it's a bacteria and it helps us to fix or to accumulate nitrogen into the soil. So it is used as nitrogen fixation bacteria. Okay, and it is done in the form of leguminous plants. Okay, now second, the decays of plants and animal waste. You know, this decays of plants and animal waste, this decomposes. I have told you about this decomposers bacteria. They used to feed on it and they used to eliminate or deplete this amount of waste that is available in our environment. So, and also it produces humus which increases the fertility of the soil. So, it is also useful in agriculture. Third, let me write down for a second, decay. Decaying means the bacteria decompose the dead animals and plants remains. Decay of animals and plants waste. Okay. The next is food production. अभी हमने एक एक्टिविटी डिस्कस की है जिससे हमने मिल्क को कर्ड बनाया, and how this milk is converted into curd with the help of bacteria. So food production में भी आपको बैक्टीरिया बहुत हेल्प करते हैं. Okay, so food productions, the bacteria that is used is called lactobacillus bacteria, which helps in the process of fermentation and production of yogurt. So which bacteria help you form milk into curd? It is lactobacillus bacteria, and it is come under the category of bacillus shaped bacteria. You know, this bacillus shaped bacteria causes many type of diseases, but it is also useful for making curd also. So every organism has its goodness also, its badness also. Okay, so bacteria are used in food production also. Fourth, digestion in animals. You know, in our human intestines and also animals' intestines, there is a bacteria reside there. हमारी intestine में एक bacteria रहता है, and the name of that bacteria is E. coli. Okay, or full form Escherichia coli. This Escherichia coli is digested in our, or it is present in our intestine. It helps in the digestion of our food. So it is also available in herbivorous animals and as well as human body, which helps in the digestion of food. Next, fifth, these bacteria are used in our industries also. Let me write down industries and we will discuss where this bacteria is useful in industries. You know, this meat, you know, the leather, how this leather is made, this bacteria is produced there on this meat, and meat is become tender. That meat को हम tender, that is soft बना लेते हैं, and that's why the tanning होती है skin की वो अलग हो जाती है. So tanning of skin is done and we get leather out of it. So this is the one of the use of bacteria in industry. The next use, it is help to produce wine and vinegar. How wine and vinegar? By the process of fermentation. Next, it helps into the production of industrial chemicals and citric acids. ये आपको इंडस्ट्रियल के जो केमिकल्स होते हैं और सिट्रिक एसिड होते हैं उन्हें बनाने में भी हेल्प करते हैं बैक्टीरियाज. नेक्स्ट, they helps in the production of some vitamins and antibiotics. Okay, so because you know E. coli bacteria, lactobacillus bacteria, they are also good, so they helps in the production of some vitamins and some antibiotics. You know, linen fibers, how you get linen fibers? You know, this flax plant from which we get linen fibers by a process called retting. What is retting? The bacteria are entered into the stem of this plant and destroy the stem tissues. जो stem के tissues होते हैं उन्हें वो destroy कर देते हैं और जैसे ही वो destroy करते हैं जो पूरी stem होती है वो fibers में convert हो जाती है और we get strings out of it, that is threads मिल जाते हैं और इससे आपको linen के fibers मिलते हैं. So आपको industry में retting process से fibers मिलते हैं linen के, that is threads मिलते हैं जिससे हम लिनन फाइबर बनाते हैं. दैट इज आपके जो क्लोथ्स बनते हैं ना लिनन क्लोथ्स, आप आपका मम्मा से बुला सकते हैं और मंगा के देख सकते हैं कि लिनन क्लोथ किस तरह के होते हैं. सो दिस वाज द यूज ऑफ बैक्टीरिया. नाउ दिस वाज द यूज ऑफ बैक्टीरिया इन अ गुड सेंस. नाउ some bacteria are harmful for us also, and we have discussed some bacteria that is they causes diseases to us. They also spoil food to us, that is spoilage of food is done by bacteria. We may be having some disease due to bacteria like tuberculosis, typhoid, tetanus, leprosy, chlorea. So different types of diseases can be caused by the bacteria to us. Okay, now we will be discussing about, as we have discussed about nitrogen fixation. Okay, now we will be discussing about a cycle called nitrogen cycle. Nitrogen cycle. Now, this nitrogen cycle, you know, the nitrogen is present in our air, is the most abundant gas that is present in our atmosphere. That is 78% of nitrogen is there. Now, we know that plant cannot directly use this free available nitrogen that is available in our atmosphere directly. The rhizobium bacteria present in the roots nodules of the legume crops convert this free nitrogen into ammonia, and this ammonia is used as a fertilizer by the plants for their growth. Now, this conversion of atmospheric nitrogen to ammonia is called nitrogen fixing, and the whole process of replenishing, that is nitrogen, free nitrogen is created into ammonia. This ammonia is used by the body, plants, and this plant is eaten by us. Then we die, or the remains or the waste of our faeces waste of us or animals is deposited on the soil, and then we have denitrification bacteria which convert this ammonia into nitrate, free nitrogen, and this free nitrogen is released into the oxygen again, and this whole process is called nitrogen cycle. Please refer to the image given in your book of nitrogen cycle. So you see that this nitrogen is converted into nitrogen fixing bacteria. With the help of nitrogen fixing bacteria, that is rhizobium, it is converted into ammonia. Okay, now this ammonia is used as a fertilizer by plants, and this plants is eaten by this cow. The cow is given, okay, this cow is eaten by this plant, is eaten by this cow. When the waste of this cow is given to the soil, then again the process of ammonia is given to the soil again, and then the accumulation of nitration is given. Now, with the help of denitrification bacteria, this free nitrogen is again created, and it is again go to the atmosphere, and whole cycle goes on repeating itself. And this is the process of nitrogen cycle. Now I will be telling you about that how this nitrogen free nitrogen can be converted into ammonia with the help of lightning. And one more fact site is given, that is this bacteria, it is helpful in genetic engineering also. Some person or some of your parents may be suffering from a disease called diabetes. What is diabetes? In which the pancreas of the patient is not able to produce very little or no insulin. So this bacteria, okay, this bacteria is used to create insulin artificially, which is helpful to treat diabetic patients. Okay, that will be all for today's class. Now we will be discussing next topic.